Hey, this is Matt. I always start out like that, don't I? Hi, this is Matt. What do you think? Vote on www.vote for bad intros from algaeresearchsupply.com. Hi there, this is Matt Huber from Algae Research Supply, and we are going to be talking today a little bit about instrumentation so that we can better communicate with our algae cultures. So let's just jump right in. Algae cultures, they live in water. And so what we need to do to be able to observe those things is to think about the culture in three steps: the physical, chemical, and biological. I'm hoping to put little tabs up there later on. Physical, chemical, and biological aspects of the water. The physical environment includes things like temperature, which we use one of these very high-tech thermometers for. This is just a regular old spirit thermometer. You can use digital thermometers, fancier, whatever you need to do. So with temperature, you use a thermometer to control the heat, but you can then use a heater to keep the heat where you want it. For spirulina, you want to be in the at least 80 to 85, up to around 90 to maybe close to 100, but you don't want to go any over 100 because the growth rate is going to slow down there. So heaters are really great for that. So physical environment includes temperature. It also includes some salts. So that's why in all of our spirulina kits we include, and any of our algae kits, we include modifications to water. So these are algae culture salts. They consist, depending on the culture that you're growing, it's going to contain sodium carbonate, or for the spirulina, it's sodium carbonate, sodium bicarbonate, sodium nitrate, potassium nitrate, let's see, sodium chloride, and what other things do we have in there? And then some trace metals. But the trace metals are also included inside of the nutrient mixture, which kind of borders now onto the chemical side, and we'll get to that in a minute. Other things about the physical nature of the thing. So remember, we're looking physical, chemical, biological. In the physical, you need mixing. And that is why we encourage you guys to mix your bubble or mix your cultures with aeration. Air is a great way to get everything to move around the culture. So we strongly recommend that. Aeration also helps out with some of the chemical side, which is next. When you put a volume of air into water, it's going to do what's called mass transfer. So we're transferring a mass, believe it or not, of oxygen out of the culture. That way the oxygen doesn't build up. In a good, healthy algae culture, you're going to get up to 5 times the concentration of the atmospheric oxygen in your water. So that means that there's going to be a lot of O2 that could become toxic, believe it or not, to your algae culture. So you want to get that out. Driving CO2 in, not really as big of a deal especially for the spirulina culture, because the salts that we provide have a solid form CO2 in the form of sodium bicarbonate, okay? So that solid CO2 acts not only as a buffer, but it also allows for the release of CO2 into a bicarbonate, both which are usable by the algae to do photosynthesis. Okay, so other chemical things. They include the nutrients, and this is one of our small nutrient bottles that comes in our spirulina kit, and all of our kits look something like this. So these nutrient containers have the main macronutrients, which include N for nitrogen, P for phosphorus, and K for potassium. That's NPK, as well as they have trace metals, which are the smaller amounts of nutrients that you need: manganese, iron, molybdenum, let's see what other crazy stuff in there, cobalt. All those things are needed in very, very minute quantities, and those are all contained in our nutrients. So that's kind of the chemical side. What other chemical things we have in here? Water, of course, is one of the chemical parts. It's both a physical environment, but it also does a lot of work, or is chemically used by all organisms on the planet. And pretty much that brings us to now the biological. So we have here one of our fine algae cultures with a little light behind it, so it looks cool. this is where we introduce the biological to our container. So in all of our culture kits, we have we include an inoculum. This is Arthur Spire here, which is spirulina. And that spirulina then you add, so you would add your salts and your nutrients to a bottle of water, get out all the dissolve, and then you can take your water then and pour it into a tissue culture flask, and then add your inoculum right to this flask, and it's going to start growing. But when it comes to the biological, this is what your goal target organism is to grow. So you're trying to make this one grow as well as it can. So one of the things you want to do is you want to eliminate all of the other possible organisms that could grow in there, which is why when we speak of biology, we want you to use bottled water, because the bottled water in the factory, we like spring water. The bottled water from the factory goes through a process of disinfection, whether it's by UV or ozone. The water that gets put in here, generally speaking, doesn't have a lot of organisms growing in it, which means that when we introduce a whole bunch of cells of algae, then that's going to be the dominant organism inside that container. Alternatively, if you want to clean things out, if say you've got some mason jars or some Erlenmeyer flask or a beaker, or even a used bottle of water, these plastic bottles can be used over and over again. You want to clean it out. So to clean it out, make sure you use soap and water and rinse it really well. So, my test for this in my lab is I use regular dishwashing soap, and then I rinse everything out. I have a little hose, and I rinse everything out, and then smell the container. Your nose is a very sensitive instrument. If you still smell soap, continue rinsing. So that's pretty much it. We've got the physical, chemical, and biological components that make up the ecosystem and the habitat for the for microalgae. Oh, one other thing too, I completely forgot on the chemical. pH probes. These are pH, the negative log of the hydrogen ion concentration, is very important chemically to make sure that you're in the right range. Spirulina likes to be at around a pH of 10. Other organisms that are freshwater like it closer to 7. Seawater has a pH of 8. So that's really low. Everything's dead. The pH of 8. It's actually the oceans right now are 8.2, but when we're not in coronavirus lockdown, there's a lot of pollution. CO2 in the atmosphere makes acid in the ocean. So we're acidifying the ocean. The pH is slowly dropping. Anyhow, we'll save that for another talk. And one last note then too on the biology side of it. If you want to quantify how much biomass you have in there, kind of nice to use one of our Seshi sticks here so that we can quantify the amount of algae that we have in the container. All right, that is just about it for our physical, chemical, and biological properties. Thanks so much for coming by. Hello, who do we have here? Ah. Hi, can you hear me?
Yes, this is Ramil Aguda, University of Louisiana.
How are you?
Good.
I'm going to turn the volume up on this thing right here.
I've been using your F/2 media for maintenance of my culture, and it's been doing great. So,
thank you.
And we also use some of your algae for demonstration of the photobioreactors.
Cool.
When our sponsor came over to see what we're doing with our algae systems.
That's awesome. It's glad to finally talk. I know we've spoken a couple times in the past. Thanks for your time on Facebook, I think it was yesterday. That was awesome.
All right. And every year, like for the past 2 years, we also have hundreds of high school students see our algae.
Oh, really?
Growing systems. So definitely it's good for science education.
Well, thanks. We're really pushing that. So, we're a couple questions. We're the only ones on. I did a quick little physical, chemical, and biological video just now, but um, uh, it's just you and I in the recording or in the video right now. So tell me what you guys are doing down there, and how we might be able to help out going forward.
Oh yeah, definitely. So we have three studies going on.
Huh?
One is how does algae actually feed on wastewater? There's a lot of studies done on it
Cool.
already. I compiled possibly 20 to 30 studies on a certain strain of algae that's from secondary wastewater treatment. So it's partially treated human wastewater, municipal wastewater treatment.
Acidic edesmus then?
No, this is Chlorella vulgaris.
Oh, really? Okay, awesome.
And then I also have another study where we're measuring how much oxygen the algae is producing. And that's quite a challenge because nobody has done it.
Really?
Mm-hmm. So there's a lot of studies on CO2
Uh-huh.
fixation. You know, how does algae use up, how much CO2 algae uses up. I read your master's thesis. So yeah, the marine algae study with the bicarbonate carbonate system. So I've been trying to figure out right now how to actually measure oxygen production.
Oh, that's really easy. And I did oxygen production as a metric for CO2 or for photosynthesis.
The synthesis. Mm-hmm.
Right. And that was and basically what you want to do is you want to normalize the new mass of oxygen you produce to something, whether it's chlorophyll or grams per liter of dry weight. You need to normalize that and then do that as over time. So it's milligrams of oxygen per gram of carbon per day.
Mm-hmm. Yes, that makes sense. It does because, you know, carbon dioxide consumption and oxygen production is proportional to the cell concentration.
Right, right.
Chlorophyll, chlorophyll amount or concentration.
Yeah. So if you
Okay.
normalize that to how much oxygen have you, I haven't, it's been a long time since I've looked at those numbers. And the reason I'm thinking that too is that we've got a lot of people that ask questions about that. They're trying to do little projects. Matter of fact, I had the Pasadena astrophysics laboratory class come in here a while ago, actually to my lab, and they were trying to do some life support system stuff for space travel. And I'm a huge science fiction fan, so that's one of the numbers I want to go find out is by dry weight of Chlorella vulgaris and or spirulina, which is that guy there.
Those are the two species that I'm working on too.
Okay. Are you getting the spirulina to grow in your wastewater?
Yes. But for now, we're only doing chlorella. Unfortunately, all our labs are shut down, so all the algae is dead.
Ours are still up. So
Your
when you guys are ready for some more, we'll ship some out to you. Get you started.
So yeah. So what my plan is to just really, well, I did cryopreservation.
Okay.
Mm-hmm.
That should be good.
Else, yep. Or else we'll just get more media from you, and then
Yeah.
this collection center in Texas
Yeah.
is giving away a discount for strains of algae which have been banked in there. So that's good. And then the third one is I'm also working on attached growth reactors. So we're doing plates stacked on top
When
of
you
each
talk
other.
about that, are you talking about like a what is that? Something bed reactor, like the a turf algae turf scrubber kind of a thing?
Yes. Good memory.
This has been my world for the past 20 years.
Oh my, I'm talking to the expert. So
I'm one of many, but thank you for the compliment.
where are you based at, Matthew?
We're down in San Diego.
Okay. Oh, the best place to do algae work.
It's all right. The rent's a little high sometimes, and that's why we're actually operating out of my garage right now.
Yeah, when I checked your business address, I'm like, Oh, it looks like a home-based business.
It is absolutely a home-based business.
Well, you could really do it actually in the comfort of your home, and you're following stay-at-home order.
Relative comfort. There's two I'll call them students inside right now
Uh-huh.
that make it a little bit more challenging. But
Okay, that's good.
yeah, well, that's great.
Um,
So you're doing wastewater work, you're doing oxygen production again with wastewater, and then algae turf scrubber stuff with wastewater. Is that correct?
yes, that's the plan because just like what you said, there's a lot of aerospace research stations who are interested in growing algae in outer space. It has been done, but we're doing it in a way that we're very comprehensive with what we do. And those are areas that haven't been explored,
Yeah,
yeah, for space explorations. But for now, for Earth, back to Earth, in reality, no one actually is interested in looking at how much algae could grow in municipal wastewater because their major concern, and this is what my professor in wastewater treatment, he's an expert in that,
hmm,
the solids in the algae
uh-huh.
treatment pond is a major concern when you actually release it to rivers or lakes.
Yeah, they can go back.
Suspended
One of the
solids.
one of the things that I've been looking at and that I'm trying to solve is, and this might be a point of collaboration. Algae is really great as a biostimulant and soil amendment for
Yes.
for crops. And
Mm-hmm.
so right now I'm working with a couple different groups that are considering doing R&D on that and putting it onto their fields for organic production. Um, so are you guys looking into that as well?
Not at the moment right now because it's all laboratory scale.
Right, right.
But the second concern right now is there's not a lot of studies on oxygen production. I just found one paper on a research group in Brazil was done algae oxygen production. That's it. The rest of the other 45 publications that I've been compiling so far because of stay-at-home orders. That's what I do. Just read and compile information from
Hey,
the.
well, you know what? That's something that I've been looking to do anyhow. I've got an oxygen probe here, and I can plug it into the computer, and
Mhm.
I could get that. I could get a culture of chlorella humming, which I need to do anyway, and I can at least get on the easy. I could use a Seshi stick, and I
Right.
can get a Seshi disk depth to the oxygen production.
Right. So you could correlate biomass cell concentration with oxygen production real time.
Yeah, pretty. I think I could do that pretty easily. Spirulina should be the same way, and then of course you have to do that across light regiments. So you're
Hmm.
doing basically a P versus I curve. If you're familiar with those,
Oh, are you talking about spectrophotometry?
no.
No, optical
Oh,
density or
go online. I've got something called on the brainy briny instructions. Look for
right.
creating a P versus I curve, photosynthesis versus irradiance. So
Okay.
basically, looking at oxygen production
Mm-hmm.
per volume of water based upon the light intensity. So
Oh.
in your pond, what you're doing is you're integrating all those light intensities down to your 1% light level, which is when it's
Mm-hmm.
concentration intensity. So what we would do, or the way you do that to estimate it in the laboratory, is you would start at super bright or super dim, and then you just slowly add a little bit more light to the system, and then wait for your culture to stabilize with oxygen.
Mm-hmm.
So I did all this literally on a strip chart recorder with paper, and then I had to get a ruler out and measure the slopes by hand.
So I
So
know
rolled up paper.
I've read your yeah, it's written in your algae culture manual that whenever I scale up, I should start with dim and then over a few days I should increase my light intensity.
Right, right.
Somebody's using your culturing manual, that's me.
That's
So
Yeah, no, the dim light helps out because when the cells are when there's not very many of them, it's too bright oftentimes,
Mm-hmm.
and we don't want to oversaturate the. So you're familiar with saturation of the photosynthetic apparatus, right?
Yes, yes.
Okay, so that's what you don't want to go over by too much, uh,
Right.
giving too much light, because then they're going to be wasting on energy to get rid of the energy. They're going to be using it
Mm-hmm.
to get rid of the energy.
I
So
see.
that's what's damaging. So they've got to go into photoprotective modes, get in their xanthophyll cycling pigment up. They've got to down regulate different parts of photosynthetic process, and it just makes it a big hassle for them.
Have you optimized it for chlorella? Have you optimized the light for chlorella and spirulina? Because I
Yeah,
just want to use a par meter.
20 centimeters depth with sunlight is about what you want. So you want to have use your Cecitus depth. You got to have some dark for the plants, but if you're talking ponds, which I believe you are,
Mm-hmm.
I think the saturation intensity for chlorella is going to be around 120 microeinsteins per meter squared second. But that means that as you integrate that depth over a pond, you're talking the surface of the pond is going to be around 2,000 microeinsteins, because that's sunlight at noon on a summer day. And then you get down to your 1% light level, and that's going to probably be in between 3 to 4 centimeters of Cecitus depth, which means that's now your Cecitus depth times 3, which gets that 4,812. That means that your 15 of your 20 centimeters now is at in the light. So your 1% light
Mm-hmm.
is down at 15 centimeters, which means that there's a couple of that centimeters down there at the bottom that are in darkness, and that's fine because it's going to mix.
That's good. Because later on, we have several designs that we're 3D printing for turf filter. And definitely that's also using less water. So um, that's also something to try and do some measurements and comparisons with ponds.
Yeah.
Um,
Well, cool, man. Well, I'm out of time. I can only go for 30 minutes on this. But hey, it was great to chat with you. Let's stay in touch, and I'd love to hear what you guys are doing with your students and the education outreach there. But I do like chlorella and spirulina, two of my favorite organisms. So feel free to reach out and ask any questions.
oh yeah, I'm writing proposal to the National Science Foundation on algae,
Huh?
and director for the bioengineering program was interested with algae viability studies. So also, if any proposal would always have an outreach component. So
Okay.
definitely we'll keep you in mind.
Yeah, and if there's any private company work that can be done too, we are always looking for new sources of revenue. We've got to bring on. I'm looking to hire a new algae culture person as soon as we come out of lockdown. Provided that we have the business. Let me know if there's anything we can do to jump on with your project as a for-profit company. So
Right.
I don't know if there's any possible public-private stuff that you guys could do, but we'd be happy to work with you on that.
Mm-hmm. One of the things that actually would be interesting would be biofuel production, but I know you've been there, done that.
Biofuel production let's look at this right now. A gallon of biofuel would cost at least $300 a gallon to make scale, and oil right now is negative $5 a 55-gallon drum right now. So
right.
there's no market for it. There's
no
market.
Yeah, we gotta go for high value like nutraceuticals or
nutraceuticals, even oils, DHA, EPA, ALA, fish food, even soil amendments, all those things that are easy to do that can help out. That's what we need to hit, in my opinion.
Mm-hmm.
But
how about proteins?
Proteins can help out too. Yeah, algae can be if you do algae as a feed or as a feed supplement for cattle, they part less currently. So methane goes down. So that means that you're actually doing climate change work by giving cattle some algae.
Yeah, that's true. That's a major source of greenhouse gases.
Yep, outside of my children.
Oh, wow.
Water. Mm.
I feel like I talked to you a lot regarding all this almost 4 semesters of studying and
Yeah.
thinking, and dreaming about algae, and you're like the right expert to have this discussion.
Well, thanks. Yeah. So are you a PhD student or a master's or?
Yes, I am. So I'm aiming for 4 publications for the algae work.
Oh,
It's
that's
multitasking, but definitely, yep. I'll keep in touch with you because this is something we could move forward with. Whatever research data I have, I'm willing to open up for supporting your business.
Oh, sure. I'll even scrutinize your research data as well if you want me to.
You can
Provided
be
it's in
a
little
reviewer.
chunks that I can digest in a reasonable amount of time. Because as my operations manager, who's off camera here, is probably thinking, yeah, send Matt something to review and read. You'll get it back real.
All right. I'm supposed to have 4 undergraduates here
Wow.
this Zoom meeting, but they're all busy with final
All
exams,
right.
and
Well, I've got to get going
I
anyhow.
apologize. But thank you so much for your time.
Sure thing. You take care. Thanks for calling in.
All right. Thank
Okay,
you.
bye now.
Bye.